Hello, my dear students. Welcome to the microbial world, invisible wonders of life. This is Dr. Syed, Assistant Professor from the Department of Studies in Microbiology, Karnataka State Open University. In this lecture, let us discuss the introduction of microbiology, historical events of microbiology, different branches of microbiology, and the contribution from the scientific community in the field of microbiology. Microbiology is a field of science which deals with the study of microorganisms. Over the years, the microbiology has rapidly expanded its roots to become one of the important scientific domains. Microbiology can be grouped based on the type of study. For example, the study of bacteria is called as bacteriology, the study of fungi is called as mycology, the study of algae is called as phycology, the study of parasite is called as parasitology, and the study of viruses are called as virology. Along with this, based on the fields and applicative properties, microbiology can also be classified as medical microbiology. It is a study of microorganisms which are important in the field of medicine. There are large group of microorganisms which are said to be beneficial in the field of medicine. For example, the group of actinomycetes are capable of producing antibiotics. There are various bacteria and fungi which are capable of secreting value-added products which are having biological activities such as anti-cancerous, anti-diabetic, antioxidant, etc. Along with this, there are certain group of microorganisms which are capable of causing infections and diseases. Some of them are coronaviruses, which caused COVID-19, the recent pandemic, and Mycobacterium tuberculosis, which causes tuberculosis, etc. Similarly, environmental microbiology. It is a study of microorganisms which inhabits in various environments such as soil, plants, water, etc. Here, the group of microbes play a very important role in ecological system. For example, Acetobacter, Azospirillum are known to fix atmospheric nitrogen and help the plant for their growth and reproduction. Similarly, there are a group of symbiotic microorganisms, especially in the rhizospheric soil, which form a symbiotic association with the plants and protect them from invading pathogens. At the same time, there are certain group of microbes which are known to cause plant infection, such as examples for these Xanthomonas species, Fusarium oxysporum, tobacco mosaic virus, etc. In a similar front, agriculture microbiology. It is a study of microorganisms which are associated in the field of agriculture. There are a group of microorganisms which are known to act as a biopesticides. Examples for this, Pseudomonas fluorescens. Pseudomonas fluorescens is a gram-negative bacteria which are widely used as biofertilizer and biopesticides. They provide plant growth promotion to the crops and enhance the crop yield. And similarly, even in agriculture microbiology, there are a group of microorganisms which are known to infect the crop. Examples, Phytophthora infestans, which causes late blight of potato. Similarly, industrial microbiology. It is a study of microorganisms which are industrially important. There are large group of microorganisms which are capable of producing antibiotics, enzymes, food products, vaccines, amino acids, chemicals, etc. Examples: Acetobacter, which is known for its production of acetic acid; Saccharomyces cerevisiae, which is known for the production of alcohol; beta glucosidase, the enzyme which is obtained from aspergillus; amylase enzymes are obtained from species of Bacillus subtilis. Moving on to food and dairy microbiology. Food microbiology is a branch of microbiology which deals with the microorganisms which are associated with the food. The best example is mushroom what we consume. It comes under fungi and it is said to be high in nutrition. Similarly, dairy microbiology. There are certain microorganisms which play a very important role in the dairy products. And due to this action of these microorganisms, most of the dairy products are formed. Example, lactobacillus. So, we just now discussed about the microorganism. But are you aware that it is believed that only 3% of microorganisms have been discovered? And what about the rest? They are waiting for people like you to discover. So, moving on to the size of microorganisms. Microorganisms are invisible to the naked eye. They require a specialized instrument called microscope. Here you can see the size distribution with respect to eye, light microscope and electron microscope. So usually microorganisms are 100 micrometer or below. Hence they require microscope for their to see their morphological characteristics. In this slide we are seeing the first microscope which was constructed. Here you can see the magnified lens along with the specimen holder where the microbial sample was placed here, a screw for the adjustment and the handle. This first microscope was constructed by Anton van Leeuwenhoek. He was the first person to observe and describe microorganisms accurately, and he named them as animalcules. Anton van Leeuwenhoek is also considered as the father of microbiology. He took high quality lenses, magnified lenses, and constructed the microscope, and he was able to isolate microbes from different sources such as rainwater, pond, well water, human mouth, human intestines, etc. So in this lecture we will be discussing about the scientific events from various scientists who have contributed in the field of microbiology. Before that, let us discuss the historical events or the hypothesis in the history of microbiology. Spontaneous generation. During those days, before the advanced scientific knowledge could evolve, there were a group of scientists who believed that life originated from a non-living matter. For example, if you have dirty materials along with the nutritive substance, after 21 days you can see the mice. According to Aristotle, he stated that there was some vital force contained in a given organic matter that can create living organism. In simple words, we can say that spontaneous generation stated that living organism arise from non-living matter. But there were a group of scientists who disproved or disagreed this spontaneous generation. One among them was Francisco Redi. He was an Italian scientist who disproved the concept of spontaneous generation with his observation and with his experiment. He conducted an experiment wherein he took 3 jars. In jar number 1, he added a slice of meat, which was opened, and in second jar he placed a slice of meat and he covered with a netted material wherein the air could easily pass through this. And in jar 3 he covered it, he sealed it where air cannot pass through it. Later he observed that there were development of maggots on jar 1 and development of maggots in jar 2 as well, but whereas there were no maggots in jar 3. He concluded that it was the fly which was carrying the maggot, which was the source for the maggots, which laid egg on the meat layer, which resulted in appearance of maggots. Further, John Needham revived the theory of spontaneous generation. He took a meat broth or meat infusion and he heated. Then he cooled and kept it open, and after a few days the meat broth or meat infusion was contaminated with microorganism. Later, in order to disprove the spontaneous generation, Spallanzani conducted an experiment wherein he followed the Needham's process but in a different manner. He sealed the jar containing meat infusion or meat broth and heated, and he cooled. The sealed jar was kept for longer period of time with no microbial growth or no contamination. Further, in order to disprove the spontaneous generation, Virchow, the German scientist, challenged the spontaneous generation with his concept of biogenesis. According to him, he stated that living organism arises from pre-existing life, whereas Virchow was not able to convince the scientific community with his backup convincing experiments. But later in 18th century, the French scientist named Louis Pasteur demonstrated that microorganism came from air, which was the responsible for the contamination of broth. He is also recognized as the father of modern microbiology. His inventions in the field of microbiology are still being practiced in the modern days as well. He conducted a swan neck experiment wherein he added a meat broth or meat infusion in an apparatus or a glassware which was specialized with its structure, and it had a unique structure of like a swan neck, where the bent shape was heated along with the meat broth and kept opened, but the broth was sterile without any contamination. Once this neck was broken, the broth was contaminated, which resulted that the air which was passing through this opening was trapped in this specialized structure which was bent and which was not reaching the broth. All the microbes from the air were trapped in this bent structure. So this experiment is called as swan neck experiment. So moving on to the contributions of Louis Pasteur, he demonstrated that the fermentation was due to the actions of microorganisms which made wine sour. He also invented pasteurization process which is still in practice. Pasteurization is a process for killing unwanted microorganisms by heating the liquids sample at a specific temperature and specific time. He also worked on creating vaccines for rabies, anthrax. He also proposed that life existed from pre-existing life. He also contributed for the development of sterile techniques which are followed in hospitals. He developed a steam sterilizer, hot air oven, and autoclave. He developed the liquid media concept. He used nutrient broth to grow microorganisms. Nutrient broth is nothing but a nutritive infusion which supports the growth of microorganisms. He also worked on germ theory along with Robert Koch. Joseph Lister. Joseph Lister is well known for his antiseptic medicine and he was pioneer in preventive medicine. He is also called as father of modern surgery. He used carbolic acid as an antiseptic agent to prevent surgical wound infection. And he also contributed the technique of pure culture and worked to prevent bacteria entering the body. He also developed carbolic spray and machine to diffuse carbolic acid into air to prevent contamination. Edward Jenner. He is referred as father of immunology. He was the one who developed smallpox vaccine. During those days, smallpox was one of the major concern across the globe. He noticed that milkmaids don't get smallpox. He stated that the pus in the blister which milkmaid received from the cowpox protected the milkmaids from getting smallpox. During those days, milkmaids who were milking the milk from the cow were protected with the smallpox infection, and he noticed that and he developed a solution containing the blisters of milkmaids and he injected that formulation to a 8-year-old boy who was protected from smallpox and he developed an immunity which led to the development of smallpox vaccines. Paul Ehrlich. He was pioneer in the field of hematology, immunology and chemotherapy. He discovered magic bullet salvarsan 606 to cure syphilis. He is also referred as the father of chemotherapy. And he was the person who applied stains to the cell and tissue to study their function. He introduced the chemical formula atoxyl to treat sleeping sickness, and he also received Nobel Prize for Medicine in 1908. He was the person who developed staining procedure for Mycobacterium tuberculosis. Later, one of the important scientists, Robert Koch. Robert Koch discovered the bacteria anthrax bacilli, cholera bacilli, etc. These were the pathogenic microorganisms during those days which were known to cause infection in human and other animals. Koch also observed the phenomena of acquired immunity. He was the first person to introduce solid media for culture of bacteria. The media here refers to the nutritive components which are infused along with the agar to give a solid consistency. He also developed the pour plate technique for the first time to culture the bacteria. Robert Koch proposed that every disease caused by a pathogenic microorganism which infect an individual, and when you isolate the microbes from the infected individual, you can culture the microbes, the pathogenic microbe in a pure form. And when the pathogenic microbial colony is injected to a healthy organism, the healthy organism was reported to develop the same disease. And when you isolate the pathogen from this secondary infected organism, the same pathogen is obtained. So these are Koch postulates which he proposed. He also described the hanging drop method for testing the mobility of the bacteria. So these are the four Koch postulates which he proposed. Another great scientist, Alexander Fleming. He is recognized for the discovery of penicillin, which saved millions of lives. He was able to isolate a mold, that is fungi, which was later identified to the member of Penicillium genus. During those days, even a small wound could be a life-threatening, and people used to die due to the infection. And Alexander Fleming was a scientist who was working on development of antibacterial agents, and he discovered that the Penicillium species was able to suppress the growth of gram-positive bacteria, and he was really inspired with this result. He cultured that fungi and he named it as mold juice, the broth of that fungi, and he was the person who discovered penicillin. And along with this, he discovered lysozyme enzyme, and he reported the presence of lysozyme in mucus, tears, egg white, saliva, etc. Along with this, he noticed that lysozyme was able to dissolve the bacteria, and he was the first person to predict the emergence of drug resistance with the overusage of antibiotics. And drug resistance is said to be one of the major concerns in recent times. Martinus William Benrick, he developed the enrichment technique to isolate a group of bacteria. Among different colonies, he developed a technique which helped to isolate a particular bacteria from the group of different bacteria. Also, he was the first person to isolate sulfur reducing bacteria and sulfur oxidizing bacteria from the soil, and he isolated free living nitrogen fixing bacteria Acetobacter from the soil, and he was the person who confirmed the tobacco mosaic virus was the causative agent in plant, wherein the virus was able to inject inside the host and was capable of reproducing with the help of host plant. Sergei Vygotsky, he was one of the major microbiologists who reported that the involvement of microorganism in geochemical cycle, and he stated that nitrification process in the soil is due to the action of bacteria. He also found out that autotrophic nutrition of bacteria, chemolithotrophic nutrition of soil bacteria. He discovered anaerobic nitrogen fixing bacteria, Clostridium species. Hans Christian Gram. He was the microbiologist who discovered the staining technique which differentiate and classify bacteria into two groups, gram-positive and gram-negative bacteria. Gram-positive are the bacteria upon staining with gram staining technique results in blue color under the microscope. Example is lactobacillus. Whereas the gram-negative bacteria turns pink in color. Example, E. coli. This is due to the cell wall difference in the cell wall composition of the gram-positive and gram-negative bacteria, which we will be studying in our coming classes. So with this, I would like to conclude my session, which gives a brief introduction to the field of microbiology, branches of microbiology, and the important historical aspects of microbiology and the scientific contributions in the field of microbiology. In the upcoming sessions, we will be learning about the different microorganism, their reproduction cycle, their economic importance, etc. Thank you. Have a nice time.